Our today's topic is Escherichia coli. E-S-C-H-E. Escherichia coli, in short it is also known as E. coli. And this is a species of Antigenic, and this genus is named after the name after German pediatrician Theodor Escherichia, who first identified this E. coli under the name of Bacterium coli commune in 1885. So this is for general knowledge who found this Escherichia coli first time, and for exam point of view, we must understand the morphology, biochemical reactions, and pathogenesis, etc. So we start this morphology of this E. coli. Morphology of this E. coli, and this E. coli is gram negative. First of all, you must know that we have studied earlier about gram positive bacilli, and this is gram negative bacilli, or in other words we can say all rods, and they are straight rods, and they measure 1 to 3 into 0.4 to 0.7 micrometer. It's the length and width of this, and they found or arranged singly or in pairs. And most of the strains are motile by Pathogenesis flagella, and though some strains of this coli may be non-motile, but most of the strains are motile, and the fimbriae and capsules are found in some strains of this E. coli, and they are non-sporin and non-capsulated. Non-sporin and non-capsulated. So this is the morphology of this E. coli. First, they are gram negative, straight rods measuring 1 to 3 into 0.4 to 7 micrometer, and they are arranged in pairs or in singly. Or most of the strains of this E. coli is motile, but some of them are maybe non-motile. And fimbriae and capsules are found in some strains, and they are non-sporin and non-capsulating. This is the morphology of this E. coli, and now we will study the cultural characteristics of this. Cultural characteristics of this E. coli. So first of all, they are aereo and facultative anaerobes. Okay, and they grows on ordinary media like nutrient agar, and the temperature needed for their culture ranges between 10 to 40 degrees centigrade, but the optimum temperature may be 37 degrees centigrade. And when we culture on blood agar, so many strains, especially those isolated from pathogenic conditions, are hemolytic. Okay, on blood agar they see hemolytic. Okay, And when we do on McConkey mediums, this is the blood agar, okay, and and McConkey. Medium, and the colonies may be red or pink, and this is due to the lactose fermentation. Either this is due to the lactose fermentation, the colonies may be seen either red or maybe pink. And on selective medias like DCA और SS, और दूसरा है SS agar, and the growth is largely inhibited and is used for the isolation of Salmonella and Shigella, that will be studied in further lectures or videos. So when we use this selective media DCA or SS agar, and the growth is largely inhibited, okay, and these selective media are used for Shigella and Salmonella. And in general, the colonies are circular, moist, smooth, and with entire margin and non-mucoid like, unlike colonies of Klebsiella. And the Klebsiella is also we will study in next lecture. What you put in your mind, keep in your mind, that the margins and the non-mucoid unlike colonies of Klebsiella, and their colonies are mucoid, and E. coli's colonies are non-mucoid. Okay, and when we culture in liquid media or in broth, the growth occurs in uniform turbidity. एकदम एक जैसी उसमें turbidity आएगी and a heavy deposit, and which disperses completely on shaking. जब हम इसको हिलाते हैं तो ये disperse हो जाता है, ठीक है. जब वैसे तो अदरवाइज तो यूनिफॉर्म टर्बिडिटी आएगी, और जैसे ही हमने इसको शेक किया, टेस्ट ट्यूब में होता है ब्रॉथ लिक्विड मीडिया, then it will disperse. So this is all about the cultural characteristics of E. coli. And now the biochemical reactions. These all are very important for your exam point of view. Biochemical reactions. And this E. coli ferment most of the sugars like glucose, lactose, mannitol, maltose, and with this fermentation it produces acid and gas, while the typical strains of this E. coli may not ferment sucrose, है ना. सुक्रोज को ये फर्मेंट कुछ इसके स्पीसीज होते हैं वो नहीं करते. एंड द इंडोल एंड मिथाइल रिएक्शन आर पॉजिटिव, ठीक है ना. इंडोल एंड मिथाइल रिएक्शंस. व्हाट द वोगस प्रोस्कोर, इन शॉर्ट वी कॉल इट BP, एंड साइट्रेट यूटिलाइजेशन टेस्ट आर नेगेटिव, ठीक है. जिनको हम इन शॉर्ट में इंडोल और मिथाइलेट और वॉस्कोर और बोगस प्रोस्कोर BP, sorry, and this is denoted by VI, and citrate. These are the denominations for indole, methylate, bogus proscore and citrate. These two are positive and these two are negative. So these are the biochemical reactions which are seen in this E. coli. And the urea is not split in this biochemical reactions of these E. coli strains. And now we will see the antigenic structure of this E. coli. This E. coli have 3 antigens, and with additional one more addition, they are H, O and O, H, O and K, and the last or fourth one is F. These are the 4 antigens, and the serotyping of this E. coli is based on these antigens. Okay, these are the 4, 3 plus 1 is 4 antigens, and this O and this H, okay, this H is known as flagellar antigen, flagellar antigen, and O, this O is known as somatic antigen, and this K is known as capsular antigen, and this fourth one is known as familial antigens. So keep in your mind that there are 3 antigens and with additional 1 antigen, H, O, K and F. ठीक है ना? H को flagellar antigen, O somatic antigen, capsular antigen and familial antigens. And some toxins are also produced by these E. coli. So, they produce anterior toxin, hemolysin, and in this biochemical reaction, they produce 3 anterior toxin, okay, understand hemolysin, and third one is virocytotoxin, virocytotoxins. These are the 3 toxins which are produced by these E. coli. And anterior toxins, these are the anteriotoxigenic strains of this E. coli produce one or both of the 2 anterior toxins. Either it may be a heat level toxins, okay, heat level toxin, ठीक है ना? In short, they are known as LT. And second is heat stable toxin. Heat stable toxin, and they are known as ST, stable toxins. Okay. And while we talk about the hemolysin, and this hemolysin, some strains of this E. coli produce hemolysin, and which can lyse the erythrocytes of some species. तो ये क्या करता है? ये RBC या erythrocytes को लाइज करता है, मतलब उनको तोड़ देता है, खत्म कर देता है. And while we talk about the virocytotoxin or virotoxins, and it is also known as cigar-like toxin. Shiga like toxin. ओके, इसको Shiga like toxins क्यों कहते हैं? क्योंकि इसके बायोलॉजिकल प्रॉपर्टीज, कुछ फिजिकल कैरेक्टरिस्टिक्स एंड द एंटीजेनिसिटी ऑफ दिस toxin is very similar to those of Shiga toxin, which is produced by the strains of Shigella dysentery type 1, and that's why it is known as this Shiga like toxin. है ना? Shiga like toxins, and this is all about the toxins and the antigenic properties of this E. coli. Now we will study about the pathogenesis and clinical features or syndromes which are produced by this E. coli. Pathogenesis is also important for all of us. Genesis or clinical syndromes which are produced by this E. coli. And first of all, you must bear in your mind that this E. coli is a part of normal intestinal flora of man as well of animal. Okay, and there are 4 major types of clinical syndromes which are caused or produced by this Escherichia coli, and they are diarrhea, one. Second, UTI, that is also known as urinary tract infection, and third one is pyogenic infections. ओके, जिसमें हमारा पस वाले इन्फेक्शंस होते हैं, एंड फोर्थ इज सेप्टिसिमिया. These are the 4 major syndromes which are produced by this E. coli. Now we will discuss about the diarrhea, and although this E. coli is normally carried in the gut as a harmless commensal, and it may cause gastrointestinal disease ranging in severity from mild self-limiting diarrhea to hemorrhagic colitis. And there are 4 different types of diarrheogenic E. coli are recognized till now, and each associated with specific serotype. है ना? हर जो भी होगा उसका एक स्पेसिफिक सीरोटाइप होगा, एंड विच प्रोड्यूस डिफरेंट पैथोजेनेटिक मैकेनिज्म, जिसकी एक अपनी खुद की एक अपनी विशेष डिफरेंट पैथोजेनेसिस प्रोसेस और मैकेनिज्म होगी. तो देयर आर 4. वन इज एंटीरियोपैथोजेनिक, E. coli. Okay, and in short it is also known as EPEC. ठीक है ना? Enteropathogenic E. coli, and second is enterotoxigenic E. coli, and it is denominated by EPEC. Set 1, 2, and third one is anteroinvasive E. coli. Anteroinvasive E. coli, and fourth one is anterior hemorrhagic E. coli. Anterior hemorrhagic E. coli, and that is EHEC, or in second, that is also known as virocytotoxin producing E. coli. So these are the four serotypes which produces different pathogenic mechanism. Okay, and while we talk about this anterior pathogenic E. coli, and these stains adhere with tightly with the erythrocytes and leading to the inflammatory reactions and epithelial degeneration changes, and have been associated mainly with diarrhea, especially in infant and in children. Okay, इसकी वजह से छोटे बच्चे और बच्चों में डायरिया पैदा होता है. And while we talk this anterior toxigenic E. coli, and they produce heat stable introduction or heat liable anterior toxins that are also known as cholera toxins, and or they produce both, and they are known to be a major cause of diarrhea in children under the age of 5 years, है ना, 5 साल तक के बच्चों में ये डायरिया हो सकता है, स्पेशली इन डेवलपिंग कंट्रीज लाइक इंडिया. And it is the important cause of travelers diarrhea also. This is the travelers diarrhea. This one causes traveler diarrhea. ट्रेवलर डायरिया का मतलब क्या होता है? मतलब अगर किसी व्यक्ति को ट्रेवल करते समय अनफॉर्म्ड स्टूल 2 या 3 से ज्यादा बार या एक बार भी अगर होती है, और उसके साथ में एब्डोमिनल क्रैम्प्स आते हैं, नोजिया होता है, फीवर आता है और ब्लोटिंग होती है ना, तो दिस इज नोन एज ट्रेवलर्स डायरिया, एंड इट इज caused by this serotype of E. coli, that is anterior toxigenic E. coli. ठीक है. And next is anterior invasive coli, and some strains of this E. coli invade the intestinal epithelial cells as dysentery bacilli and produce disease identical to Shigella dysentery, है ना, dysentery की तरह ये symptoms पैदा कर देता है. इसलिए इसको हम कहते हैं anterior invasive E. coli, and okay, and next is the anterior hemorrhagic coli, or in other words we can say virocytotoxic produce E. coli, and these strains causes hemorrhagic colitis and hemolytic uremic syndromes. Okay, two things. One is hemorrhagic colitis and second is hemolytic uremic syndrome. And toxin responsible for this problem is known as virotoxins, because it has effect on viro cells that are produced in culture. So remember all the serotypes of this E. coli, anterior pathogenic E. coli, Toxigenic E. coli, anti-invasive E. coli, and anti-hemorrhagic E. coli, and these are the denominations which are denominated by these. And second one, we will study the UTI. And the cause of urinary tract infection, the E. coli is the commonest organism which is responsible for UTI, urinary tract infection. And this UTI occurs more in females than in males. Okay, and this is due to the short urethra and the pregnancies and the infrequent voiding and sexual intercourse, and which may lead to honeymoon cystitis. Okay, these are the causes of this UTI. And second important cause of this UTI is catheterization, and cystoscopy may introduce endogenous or exogenous bacteria into the bladder, and that may cause infection, especially in male or female. Okay, अगर हम उसमें कैथेटर डालते हैं यूरेथ्रा के थ्रू, तो उसके थ्रू इन्फेक्शन पहुंच सकता है ब्लैडर में. Okay, and that may be the cause of this UTI. And next we will talk about this pyogenic infections. Pyogenic, and this E. coli may cause wound infections, intra-abdominal infections such as peritonitis and cholecystitis. These are the two very common problems which are caused by this E. coli. And it also causes some pyogenic infections in the perianal area, or some important case or cause of neonatal meningitis. तो छोटे छोटे बच्चों में meningitis होती है, उसमें भी ये एक कारण बनता है. Okay, and now the septicemia, and this is the very common cause of this septicemia in many hospitals, or in other words, the hospital infection is caused by this E. coli, and this condition usually occurs in debilitated patients. है ना, जो कमजोर पेशेंट्स होते हैं उसमें ये अक्सर सेप्टिसीमिया होता है, और इसकी वजह से मोर्टेलिटी रेट काफ़ी रहती है. सो दीज आर द 4 things which are produced by this E. coli. One is diarrhea, second is UTI, third one is pyogenic infections and septicemia. Okay, and now the laboratory diagnosis of all these things. So first of all, we take the diarrhea, and the laboratory diagnosis of E. coli diarrhea has been a complex, as there is no single test which can identify all strains of E. coli which is responsible for diarrhea. And for this we took a specimen from feces or rectal swab, and we do the culture especially on McConkey medium, and after overnight incubation the growth on culture is identified by the colony morphology that we have read or studied earlier, and the gram staining, and motility and biochemical reactions. These are the things by which do the diagnosis of diarrhea. And in case of UTI, urinary tract infection, we took the sample of urine, ठीक है ना, और उसका जो सैंपल यूरिन का होगा वो कैथेटर स्पेसिमेन भी हो सकता है, मिड स्ट्रीम यूरिन स्पेसिमेन ही कहना में हमें लेना चाहिए, है ना, मिड स्ट्रीम यूरिन स्पेसिमेन, इसमें मिड यूरिन स्पेसिमेन MUS. ओके, ये हमेशा याद रखना चाहिए और हमें पेशेंट को बता देना चाहिए भाई तुम्हें कब टेस्ट के लिए यूरिन देना है, ठीक है ना। और इन स्पेशली इन इन्फेंट्स और इन केस ऑफ सम अनकॉन्शियसनेस जिसमें ये यूरिन नहीं होता है, तो वहाँ हम सुप्राप्यूबिक स्टेप से भी यूरिन लेते हैं, ठीक है ना। तो ये 3 कंडीशंस हैं जिसमें हम कैथेटर से यूरिन लेंगे, लेकिन हम जब कैथेटर से यूरिन लेते हैं तो हमें बैग से नहीं लेना है, सीधे हम कैथेटर से ही लेंगे। और जब हम मिड स्ट्रीम यूरिन की बात कर रहे होते हैं तो हमें पेशेंट को इंस्ट्रक्ट करना होता है, मेल एंड फीमेल को। स्पेशली मेल जब यूरिन करें तो अपने प्रेप्यूस को पीछे करें और जो उसकी जो स्ट्रीम होती है वो किसी डिब्बी या पात्र में लें, अनटच जाना चाहिए, ठीक है ना। ये थोड़ी सी ये क्योंकि इन्फेक्शन होने के चांसेस रहते हैं। वो सेम फीमेल के मामले में है, उसमें भी हमें जो मिड स्ट्रीम यूरिन, पहले का यूरिन एक पास कर लो थोड़ा सा और बीच वाला यूरिन हमको लेना है। और स्पेशली इन फीमेल्स वहाँ पे भी जो उनके मेजोरा होते हैं थोड़ा सा हाथ से उनको अलग करके और यूरिन हमें बताना चाहिए भाई पेशेंट को ऐसे यूरिन लेना है। अदरवाइज all the genital must be cleaned by soap and or antiseptic things. So this is the technique of or method of taking the specimen, especially in this urine collection. And after this, we transport the urine. हम यूरिन का सैंपल लेने के बाद में इसको ट्रांसपोर्ट करते हैं लेबोरेटरीज के लिए, और लेबोरेटरी में भी तुरंत यूरिन का